Oh, hi there. Welcome to LabTechTV. I'm your gracious host, Eva Maria. Not really, it's a used lab coat. I guess for LabTechTV we should focus on a machine that's common in almost all labs. Definitely all labs that are working with genetics or working on a genetics-based standpoint. It's called the thermocycler. The thermocycler is works off the idea of PCR. So to understand the thermocycler, common in all labs, you need to understand PCR. PCR is a polymerase chain reaction.
PCR is a scientific technique based off of the cellular process of DNA replication. DNA replication starts with double-stranded DNA. In the first step of PCR, the denaturation step, the double-stranded DNA is separated. This separation is caused by a high temperature of between 94 and 98 degrees Celsius that provides the energy to separate the strands of DNA. In the second step, the annealing step, the temperature is lowered to between 50 and 65 degrees Celsius. This temperature is required to allow the primers, short strands of RNA complementary to the DNA, to join with the single-stranded DNA. In the third step, the elongation step, the enzyme DNA polymerase, shown here as a tiny rocket man, builds new strands of DNA, one base at a time. DNA bases are the building blocks of DNA. When the round of replication is complete, there is effectively twice as much DNA as when the round started. Here, you can see that the double-stranded DNA has been duplicated. This three-step process is repeated through multiple rounds of replication until a desired amount of DNA is synthesized.
PCR is able to take sections of DNA and selectively copy those sections multiple times. So what this basically allows us to do is search DNA to see if maybe there's a sequence that we're looking for, say in a specific disease, so that we know if this person has that disease based on the genetics. Or maybe there's a strand of bacteria that's extremely infectious, and we know this because we've searched for a gene that it makes. In labs it's very helpful because it allows us to copy DNA that we're using, maybe we're transforming cells with and we need a lot of this DNA. Well, it helps us copy specific strands and then we can insert them in plasmids. So it's an amazing tool. Thanks for stopping by and viewing this episode of LabTechTV. I hope you wait for the next when we figure out what other kind of gadgets they hide in labs like these.